So thank you. Thank you to Thermo for inviting me here today. The title of my talk is slightly different. It's a proteome-based mapping of non-serous gynecological tissues. So unlike the first talk that we heard, which is very much focused on small molecules and drugs, and it's into the clinical laboratory, this is more of proteins and proteomics, and still in the research setting, but how we can eventually translate it back into the clinical lab. So just some background, as I'm sure you've heard from some of the plenary lectures that will happen today and yesterday and so on. We're in this omics revolution that's really happening right now, and genomics, as we know, is a field that's going to investigate thousands of DNA sequences, and transcriptomics is looking at the RNA. And these are really important because both genetics and functional genomics will help us understand the molecular basis of inheritance, but they cannot really infer phenotype. And so proteomics and metabolomics are fields that can use mass spectrometry to analyze the constituents that will help us understand phenotype. And that's really because proteins provide the structure and function for nearly all of life's biological processes, and I'm a protein person, so we all know some of the examples of proteins. These are like enzymes, hormones, antibodies, etc. So proteomics is quite important to help us understand biology. And so if we try to define what proteomics is, just to make sure, I'm not sure what the background is for most people here, it's the large-scale identification and functional characterization of proteins in a cell, in a tissue, or in an organism. And what's important here is that if we think about genes, there are about 20,000 protein-coding genes in humans. This we know. When we look at transcripts, there's about 50,000 transcripts because of alternate spike forms. But when we think of proteins, actually, there's at least 500,000 to 1 million proteins, so, which is largely because of post-translational modifications and the like. So you have much far more complexity in a proteome when you're trying to decipher it versus in a genome. And so mass spectrometry can really be used to study proteomics, and there's different branches that I've shown here, which includes, for example, expression proteomics. So you're looking at differential expression of proteins in a given sample. There's functional proteomics, so looking at the interaction between proteins to characterize complexes and determine its function. Of course, there's quantitative proteomics, so measuring protein abundance or its concentration. There's post-translational proteomics, studying the diverse modification that proteins can have, and structural proteomics, so the analysis of the protein structure itself. So there's such diverse applications to study the proteome using mass spectrometry. And what our field is now moving into is the integration of the omics and using it to study network biology. So proteomic information can now be used to improve existing gene models, to profile molecular phenotypes at the level of protein abundance, post-translational modifications, and protein-protein interactions, and to obtain specific pathway information using targeted mass spectrometry strategies. And you can reconstruct functional molecular networks through the integration of these high-quality information from functional genomics and proteomic data. And another really exciting field that we're moving into is what's called proteogenomics. And it's a field that's going to use proteomic information that's obtained from mass spectrometry to improve both gene annotations or the reverse. So you can customize protein sequence databases based on DNA sequencing information for that sample, or you can perform protein level validation and gene model refinement. And what this means is that you're going to have customized protein sequence databases that's generated using genomic and transcriptomic information, which can be used to help identify novel peptides, or that's not present in your reference protein sequence database from mass spectrometry-based proteomics. proteomic data. And in turn, of course, the proteomic data can be used to provide protein-level evidence of gene expression and help us refine our gene models. So hopefully, with just these introductory slides, I've conveyed the potential of this analytical tool, which is mass spectrometry, and the promise of proteomics. So for the next part of the talk, I'll really focus on one application of this tool in our lab, which is to look at ovarian cancer. So just some background. So we've talked about the background of proteomics and why we're interested in it, mass spectrometry being the main workhorse. And so today's presentation, I'll look at how we can use these tools to help us understand just a little bit more about ovarian cancer. So ovarian cancer is the most lethal gynecological malignancy. It accounts for about 5 to 6% of all cancer-related deaths. There are about over 200,000 women who are diagnosed every year worldwide, and well more than half of them will succumb to the disease each year. And the majority of these women are identified when they have late-stage disease, primarily because they are quite asymptomatic at presentation. But ovarian tumors can actually be categorized further based on their histopathological analysis. So the majority of the tumors will fall into the surface epithelial stroma, but you can also have germ cell, sex cord stromal, or mixed cell tumors. But the epithelial surface epithelial stroma can be further subdivided into these 4 subtypes. So serous ovarian cancer represents 80% of all ovarian cancer cases, and when we typically think of ovarian cancer, that's the subtype that we're talking about. That's the one that serum CA-125 is often elevated in these patients. But ovarian cancer is made up of other subtypes, and that includes mucinous, endometrioid, or clear cell carcinomas. And those 3 are what I will call the non-serous subtypes of ovarian cancer, and they make up the remaining percentage. Serous cancer can also be further subdivided into low-grade and high-grade. So what's interesting about these different subtypes of ovarian cancer is that each one actually represents a very distinct etiological disease. They simply share a common anatomical location. And what we see in this table is that the suspected etiological, their origins are actually quite different for these lesions. So mucinous carcinoma, for example, the origin is cyst adenoma. In the case of endometrioid and clear cell carcinoma, the initial precursor lesion is endometriosis, or that's what's suspected. Their pattern of spread is very different across the subtype. The molecular abnormalities that lead to the disease are quite different. Their response to therapy is very different. Their prognosis is very different. So hopefully what I'm trying to say is there's been a lot of research that's been devoted to ovarian cancer, but primarily to serous ovarian cancer and not to the other subtypes. And the other subtypes are quite different compared to serous. And so one size really doesn't fit all, and it goes back into our precision medicine topic that we're talking about. So our rationale here was that proteomic comparison of the appropriate gynecological tissues may allow us to identify subtype-specific markers. And so we want to compare mucinous, endometrioid, and clear cell carcinomas, which are the non-serous ovarian cancer subtype, to their so-called suspected etiological origins, which would be endometriosis or mucinous cyst adenoma. And so our goal was to try to identify molecular pathways that can be central to these endometriosis-associated ovarian carcinoma pathogenesis, and also to try to see if we can identify any novel serum biomarkers that are specific to the non-serous subtype to help identify these patients. And so our methodology consisted of getting fresh frozen tissue from the various subtypes or categories. So I have normal endometrium, I had endometriosis, which is the benign condition. We have clear cell carcinoma, endometrioid carcinoma, mucinous cyst adenoma, which is a benign condition for mucinous carcinoma. And so with these tissues, we digested them, did offline fractionation into 9 fractions, and then put them onto the Q-exactive mass spectrometry for data-dependent acquisition and identifying the proteome. we had over 300 fractions for this entire project that was injected into the mass spec. So if you can imagine, when you have 300 samples, it's not going to be completed in a day. This was an exercise that took many days and weeks. And when you have something that goes over many days and weeks, you have to batch them appropriately, but not to run all your controls first, because you may have batch-to-batch variation. So we did randomize all of our samples before we put it onto the mass spec. And then data analysis consisted of taking the raw files and doing max quant searches to identify what the proteins are, followed by bioinformatics using Perseus and other bioinformatics software analysis. So let's look at the data generated. I first want to start off in this figure, what you see at the top. Let me see. So I have the various cohorts, so clear cell carcinoma, endometrioid carcinoma, endometriosis, normal endometrium, mucinous carcinoma, and mucinous cyst adenoma. So these are the almost 30 different sample types that we had, and we have protein intensity here, and I'm showing you what we call the housekeeping genes, so housekeeping proteins. So these are ribosomal associated proteins, and you can see that their expression is fairly consistent and constant across the cohorts of samples that we analyzed, despite analyzing them on different days and batches. And then, of course, all the others are the other proteins that we found. So the housekeeping genes are quite consistent, but how about the other proteins? And that's really what we're interested in. So in total, and I want to focus really, because I had way too much data, I want to focus on the endometrioid clear cell carcinoma proteome here. So I won't be talking about the mucinous or mucinous cyst adenoma. In total, we identified about 9,000 proteins in these 21 samples, and there's a high correlation of protein expression profiles between the patients of the same cohorts that you can see. That's highlighted in the red boxes. But what may be interesting is a sample number 18, which really didn't group with the endometrioid carcinoma that it was supposed to be. So we chose not to include it further, but we did go back to our pathologist to kind of see if there was any misclassification of the disease, because when we included our mucinous data into this and tried to categorize it, the proteomic profiling of sample number 18 actually matched quite well with the mucinous carcinoma. And so what we found out afterwards was that EC18 was later confirmed to have mucinous-like elements by the pathologist. And so the pathology report states that there appeared to be endometrioid, but immunohistochemistry staining was more in keeping with a gastrointestinal type carcinoma, and mucinous carcinoma often has origins to GI. And so another important point for checking any proteome cohorts is really to see if we've identified any known markers. So pathologists currently make their diagnosis for these different subtypes based both on morphology as well as immunohistochemistry markers, such as some of the ones that I've listed here. And we actually identified many of the mucinous and the endometrioid and even the clear cell carcinoma markers, and we did not identify any of the serous specific immunohistochemistry markers, such as P53, WTI, or CK7. So that was pretty good news. It's consistent with immunohistochemistry. We identified known markers. And so we now looked at what is the overlap of the proteins that we identified across our different cohorts, and sure enough, there's a number of proteins that's common to them, but certainly there's many proteins that were unique to the different subtypes. And then we went on to perform what's called a principal component analysis. So this is unsupervised clustering without any enrichment to demonstrate the ability for us to distinguish the cohorts from each other, which were able to distinguish the controls from the disease, but also the clear cell from the endometrioid. And so then we decide to look further into this and try to see within the clear cell carcinoma proteome, what are the pathways that are actually enriched, and can this help us identify more information about its pathogenesis. So when we enriched for statistically significant proteins, we see that clear cell carcinoma is mainly characterized by dysregulation in the muscle and cytoskeletal related processes, whereas endometrioid carcinoma seems to be characterized by cell junction and extracellular matrix related processes. Quite nicely, it quite separates the endometrioid carcinomas from both the normal endometrium and the endometriosis. And then, of course, because we talk about integrated proteomics, and while I don't have the genomic information for the same samples that I used for my proteomic data, we went back to the literature to see, are there any so-called endometrioid-specific gene signatures or clear cell carcinoma-specific gene signatures, and there were some in the literature. So we looked to see what are the proteins elevated in those cohorts, and we see some concordance where the ones that are highlighted in red showed to be elevated in clear cell compared to, it was actually comparing it to high-grade serous ovarian cancer in the genomic data, so it's not a direct correlation that we can really look at, but there seems to be some concordance with the gene markers for these two subtypes. But that's made us think perhaps we should go back to try to understand the biology of this disease. So just to step back a little, endometriosis, it's a common disease that's characterized by inappropriate growth of endometrial tissue outside of the uterus. And there's a prevalence of about 5 to 15% in women of reproductive age and about 3 to 5% in postmenopausal women. And it's often associated with chronic pelvic pain and infertility. And endometriosis in general is a benign condition, and it's rarely fatal. It can implant and invade and penetrate into other tissues, but they generally lack the proliferative histology that's seen in malignant cells. But there's a subset of endometriosis which have these which are atypical. That's features that are not entirely benign and not completely malignant. So these atypical endometriosis is believed in the literature to be an intermediate transition between normal endometriosis and the endometriosis-associated ovarian carcinomas. And there's literature saying that these mutations in ARD1A mutations that can lead to the endometriosis-associated ovarian carcinoma, and a loss of heterozygosity of PTEN and PIK3CA have also been observed in this. So we went back to our proteomic data and looked to see if the ARIDA, D1A, and the PTEN pathways, because they're dysregulated in this cancer type, do we see that at the protein level? And we've, using Reactome, we identified key components of these pathways, and in fact, 21 of the 37 ARD1 components were identified in our proteomic data set, and similarly, we identified some proteins that's part of the PTEN PIK3 kinase pathway. So it wasn't complete concordance, so we know there are some issues here. The genomic and the transcriptomic features do not necessarily translate at the protein level, so then it made us think, well, how else can we try to identify novel mechanisms of pathogenesis if I can't go back to the literature to see what's already out there? So we thought, well, we have our own data, let's re-look at our data, and we looked at it in 4 different ways. So we have our control cohort, which was endometriosis, and we had our cancer cohort, and we wanted to focus on proteins that were completely increased in the cancer cohort compared to the control cohort, so that's the presence absence. We can look at proteins that were differentially expressed in the cancer cohort compared to the control. We could look at proteins where they were very overexpressed, but in a subgroup of the cancer cohort compared to the control, but overall they were expressed higher. Or we can look at progression, because I had normal endometrium, I had endometriosis, which is actually the precursor lesion to endometrioid cancer. So if there are markers that tend to be elevated in these groups, which I call the progression markers, perhaps they might be interesting to look at and see if they're dysregulated in our cohort. So I'll show you data from our progression markers cohort. So this is a figure that was derived from a list of about 250 proteins that actually displayed progressively increased expression from benign endometrium to endometriosis to clear cell cancer. And what we see is that it allowed us to nicely group out the endometrium and endometriosis from the clear cell, and again, kind of into the endometrioid cancer. But our N is very small. I already recognize that, and this is just discovery data that needs further validation with subsequent other samples, but it's something interesting to follow up on these pathways. And then, of course, as with anything, we do pathway analysis. We identified some overrepresented signaling pathways, in this case in clear cell carcinoma. And so I want to conclude, as we're running out of time here, that this data really represent the first and the most comprehensive study to delineate the proteomes of endometriosis and endometriosis-associated ovarian cancers. In fact, in the literature, when you look, there's very limited proteomic data on these non-serous subtypes, and none of them really comparing them to its actual precursor lesions. And our data is quite biologically sound with its expression profiles. We identified some of the major molecular pathways that's involved in this, just that gene expression data may not fully represent at the protein level, and proteomics may reveal novel mechanisms of pathogenesis that cannot be solely deciphered through genomics alone. I was going to go on to goal number 2, but I'm going to skip that and conclude just by acknowledging the members. So this is my former PhD student who's now a clinical chemistry fellow at the University of Toronto, and our collaborators and funding agencies. Thank you very much.